everyone, and welcome to Lab Week 6. This week we're going to be discussing selective and differential media. So there are different types of media. Basically, there's different types of media that we can put bacteria in to grow, and typically that growth can exist in different patterns based on how the bacteria interact with the media, either in the petri dish or in the test tube. So a common type of media is selective media. Selective media is one that is going to encourage the growth of some organisms while inhibiting the growth of others. So there's always a selection process where some bacteria will grow, but some will be inhibited. So you know you're looking at a selective media if you see words like inhibition, where some bacteria weren't able to grow. A common type of selective media is mannitol salt agar, which we do use. Mannitol salt agar, and you can tell by the name, it has a high concentration of salt in it. Most bacteria cannot survive in the presence of salt. Salt creates this hypertonic environment where it basically pulls moisture out of bacterial cells and causes them to shrivel up and die. So most bacteria, if you put them on a mannitol salt agar, they would not be able to grow. The exception to that rule is Staphylococcus. Pretty much all types of Staphylococcus, including Staphylococcus aureus, which is quite dangerous, is able to grow on mannitol salt agar. And that's because Staphylococcus can actually tolerate a high salt concentration. They're prone to growing on our skin, which has a high salt concentration due to sweat. So they actually thrive in the environment that mannitol salt agar provides. So if you put a bunch of different types of bacteria on mannitol salt agar, Staphylococcus and E. coli and Serratia marcescens, the only type of bacteria that would grow would be Staphylococcus, whereas all others would be inhibited because they cannot grow in that high salt concentration. There's also differential media. Differential media does not really inhibit the growth of any bacteria. What it does is it causes bacterial growth to occur in different colors based on how the bacteria interact with the media. So pretty much on differential media, all different types of bacteria will grow. If you put 10 different types of bacteria on a differential media, there's no inhibition. 10 different types of bacteria will probably grow. However, the growth will occur in different colors based on how the bacteria reacted with the contents of the agar. So a good example of a differential media is a blood agar. Pretty much all different types of bacteria will grow on a plate that contains blood agar, but they will grow different colors based on how they interact with the red blood cells. And we'll see this when we kind of talk about the discussion of blood agar, but depending on how they interact with the red blood cells, they will grow different colors. So to make it a little bit more confusing, plates don't have to just be selective or just be differential. Many of the plates that we work with are actually a combination of both. So you can have one plate that has both selective and differential properties. What that basically means is, if you're working with a plate that's selective and differential, it means that only certain types of bacteria will grow on the plate, but the bacteria that are able to grow will often grow different colors based on how they interact with the media. So a really good example of that is the eosin methylene blue, or EMB, agar that we commonly use. This plate is selective and differential. It's selective because it only allows gram-negative bacteria to grow. Gram-positive bacteria are inhibited from growing. So only gram-negative bacteria grow. That's the selective part. However, the gram-negative bacteria that grow, they grow different colors based on how they interact with the lactose, which is the sugar source in the media. Sometimes they'll grow this metallic green color, sometimes they'll grow almost a black color, sometimes they'll grow dark purple. It really kind of depends on how they interact with the media. So if the plate is both, only certain types of bacteria will grow. However, those certain types of bacteria that do grow will typically grow different colors based on how they interact with the media. So the very first plate that we're going to talk about is that mannitol salt agar, or MSA agar. So the main purpose for mannitol salt agar is to isolate Staphylococcus bacteria and actually differentiate Staphylococcus aureus from all the other types of Staphylococcus. There's a lot of different types of Staphylococcus bacteria, but the one that we're typically most concerned with is Staphylococcus aureus because it is the one that's most commonly known for causing severe infections. But we have things like Staphylococcus epidermidis and Staphylococcus saprophyticus just commonly found on our skin, and those species don't typically cause any severe infections, but Staphylococcus aureus is known for causing pretty much a variety of different infections. It's kind of the one bacteria that if it gets anywhere in your body, it can cause some type of an infection. So over here, we're looking at just kind of an empty mannitol salt agar plate, and typically we would inoculate with that with our bacteria and then look to see if it grew or not, and then also look to see if it grew, what color did it grow. So this plate does contain mannitol. Mannitol is the sugar source. It's what will actually be fermented, potentially for an energy source. So mannitol is just the carbohydrate or the sugar source for fermentation. Then there's sodium chloride, which is salt. And then this plate actually has something called a pH indicator, which is phenol red. The pH indicator, which is phenol red, will actually cause the plate to change colors depending on how the pH changes in the plate. So basically, the plate is kind of this reddish color right now, but if the pH would go down and become more acidic, the plate would turn bright yellow. If the pH would actually increase slightly, then that would cause the color to turn a bright pink color. And we'll see examples of this in a second. But this plate does have a pH indicator called phenol red that allows the plate to change colors depending on what the pH of the plate becomes. So mannitol salt agar is one of those plates that's actually a combination of selective and differential properties. So we're going to first talk about why it's selective. So a mannitol salt agar plate is selective because of the salt. The salt allows only Staphylococcus bacteria to grow. Doesn't matter what type of Staphylococcus. Could be the dangerous Staphylococcus aureus or the friendly Staphylococcus epidermidis. It just allows all different types of Staphylococcus to grow. So if we have a mannitol salt agar plate right here, ignore the colors for right now, but we have Staphylococcus that was put here, a Staphylococcus type of bacteria that was put over here, and maybe something like E. coli up here. We can see that the Staphylococcus bacteria grew just fine because they actually like the high salt concentration. But the E. coli that was placed up here, growth was inhibited. E. coli cannot survive in a high salt concentration because it basically dehydrates them and they shrivel up and die. So the reason why mannitol salt agar is a selective plate is because of the salt. The salt allows only Staphylococcus type bacteria to grow while inhibiting all other types of bacteria. So if you see growth on a mannitol salt agar, you know it's some type of Staphylococcus. Now, right now we don't know exactly what type until we talk about the different colors, but we at least know that we have some type of Staphylococcus bacteria that are growing on these plates, on this plate, since staph is the only type of bacteria that can grow in this high salt concentration. So we need to talk about the different colors now because we see some yellow, we see some pink. So, like I said, this plate is selective and differential. It's selective because only Staphylococcus can grow on the plate. So we know that if we see growth, it's some type of Staphylococcus. But different types of Staphylococcus will grow different colors. Some will grow bright pink, and some will grow bright yellow. So that has to do with the fermentation of mannitol. Like I said, there is mannitol in this plate, and that serves as a sugar source or a source of fermentation for energy, or ATP. So basically how it works, Staphylococcus bacteria, they grow on this plate because they enjoy the salt. If they're able to ferment the mannitol, which basically means use the mannitol as an energy source, that will cause acids to be released. As the acids are released, the pH in that plate is going to decrease. When the pH in this plate decreases, that causes the phenol red pH indicator to turn bright yellow. So when you see yellow growth on this plate, you know you're looking at Staphylococcus, but you're also looking at a Staphylococcus bacterium that has fermented the mannitol. Really, there's only one species of Staphylococcus bacteria that does ferment mannitol, and that is the dangerous Staphylococcus aureus. So often when you're looking at these plates and you see yellow growth, that's a good indication that you are looking at Staphylococcus aureus. Other types of Staphylococcus, they grow on this plate, but they don't really ferment the mannitol. They don't use the mannitol for an energy source. So if they don't ferment the mannitol, that means acids don't get released. If acids don't get released, that means the pH doesn't change. If the pH does not go down, that means the pH indicator is just going to be this bright pink color, which is what we see here. So that means we're looking at some other type of Staphylococcus, maybe something like Staphylococcus epidermidis that commonly grows on our skin. So to just kind of summarize, mannitol salt agar plates are selective because they only allow Staphylococcus bacteria to grow. However, they're also differential because different types of Staphylococcus will grow different colors on the plate. If you're looking at a type of Staphylococcus that can ferment mannitol, it will grow yellow. If you're looking at a type of Staphylococcus that does not ferment mannitol, it will grow pink. And we see these color changes because of that pH indicator. Remember, if the pH goes down, that's what causes the yellow color change. If the pH would stay the same or increase, that's what causes this kind of bright pink change that we see here as well. So the second type of plate that we'll talk about is the MacConkey agar or the MAC agar. This plate, like the mannitol salt agar, is also selective and differential. So again, what that means is only certain types of bacteria grow. However, the bacteria that grow will grow different colors based on how they interact with the media. MacConkey agar is used to isolate and differentiate members of the Enterobacteriaceae family that are able to ferment lactose. So it's a big word, but Enterobacteriaceae, that is basically just types of bacteria that are commonly found in your gastrointestinal system. So really it's just like gut bacteria. And we're looking to see if these gut bacteria can ferment lactose, which is a sugar source that they could use for energy. So MacConkey agar, it does contain lactose. That's the sugar source that'll be used for fermentation, like mannitol was in the previous one. It contains bile salts and crystal violet. Bile salts and crystal violet are quite important because they are going to inhibit the growth of gram-positive bacteria. With the MacConkey agar plate, we only want gram-negative bacteria to grow because in your gastrointestinal system, the main types of bacteria that you have are gram-negative bacteria. So when we use this plate, we want to inhibit gram-positive growth because those bacteria really aren't found in the gastrointestinal system, and we're going to inhibit them with bile salts and crystal violet. So they are what actually makes the plate selective because they inhibit gram-positives and only allow gram-negatives to grow. This plate also has a pH indicator. It's called neutral red dye, but same kind of principle. The pH indicator will change colors when the pH in the plate changes. So with this plate, if the pH goes down, the plate actually turns a bright neon pink color. And if the pH stays the same or increases, the color really doesn't change. It's just kind of the color of the media.